So I would like to begin the JSID Rising Star Lecture. The Rising Star Lecture celebrates the best emerging dermatologic science worldwide. This year's JSID Rising Star Lecture will be given by Professor Yoshihide Asano from Tokyo University. Dr. Asano's work focuses on the clinical basic research on systemic sclerosis. He has found the important role of the transcription factor FLY1 in the pathogenesis of systemic sclerosis. Dr. Asano is an associate professor of dermatology at the Tokyo University. He spent 2 years in the postdoctoral training in rheumatology and immunology at the Medical University of South Carolina in Charleston from 2006 to 2008. His lecture is titled, and Skin Research Provides a Window into New Aspect of Systemic Disease Pathogenesis. Okay, please welcome Dr. Asano. Okay,
so, thank you, Professor Morita, for your kind introduction. And I'd like to thank the organizer of this meeting for giving me this great opportunity to present at this wonderful meeting. So, can I have the slide? Okay, so I'd like to start my presentation with just a simple question. So, what's rising star in dermatology and cutaneous biology? So, what do you think? So maybe some people think that it's T17 cells in psoriasis, and maybe other people may think that it's immune checkpoint inhibitors for tumor immunity. And others may think that it's skin barriers in atopic dermatitis. And I think there are many rising stars in each research area of dermatology and cutaneous biology. But the important thing is that there are many supporting characters behind rising stars. And sometimes we can obtain a very important brand new idea by focusing on these supporting cells. And I'd like to talk about this point by focusing on my specialty, systemic sclerosis. So systemic sclerosis, SSC, is characterized by extensive tissue fibrosis of the skin and various internal organs, such as interstitial disease and esophageal dysfunction, as well as systemic vasculopathy and autoimmunity. And it's generally accepted that this disease is driven by activation of dermal fibroblasts, vascular cells, and immune cells. And the research spotlight on SSC has been put on all these three types of cells. But today, I'd like to put the spotlight on the keratinocyte and the adipocyte in my talk. So let's see the skin histology of the involved skin of SSC patients. So maybe you can see some strange cells at the dermal-epidermal junction. And for me, this cell looks dying. And this is another case, so maybe we can see much more severe change. And when we carry out tunnel staining, we can see many tunnel positive cells, which means that these cells, so there is a abnormal cell survival signaling in the SSC keratinocyte. And since 2010, there has been many reports regarding the altered phenotype of SSC keratinocyte. So let me show you some examples. So on the left side of the slide, I showed the increased expression of keratin 6 and 16, which are the wound healing associated cytokeratins, and also expression levels of IL-1 alpha and CTGF are increased in the epidermis of SSC patient, and this abnormality contributes to the activation of dermal fibroblasts in this disease. As well, there are many reports regarding the altered phenotype of various kinds of cells in this disease, such as endothelial cells, and immune cells, and adipocyte, and platelet, and myeloid cells. And this evidence suggests that there must be some critical disease factors which may integrate unique property of these cells. And if we can find such factor, we can establish a unifying hypothesis which can explain a complicated disease pathology of this disease. And as you know, the occupational SSC is an established clinical entity suggesting the critical contribution of environmental factors to the development of this disease. And consistent with this idea, the concordance for SSC is similar in monozygotic and dizygotic twins. So the molecules whose expression is altered by the influence of environmental factors may serve as a potent predisposing factor of this disease. And one of such factors is a transcription factor, FRI1, whose expression is diffusely downregulated in the involved skin of SSC patient, including keratinocyte, dermal fibroblasts, endothelial cells, immune cells, and adipocyte. And the previous reports demonstrated that the FRI1 is epigenetically suppressed in SSC dermal fibroblasts, suggesting that FRI1 may be a potential predisposing factor of this disease. And based on this idea, we decided to generate various kinds of cell-specific FRI1 knockout mice by utilizing log-speed CRISPR system. And today, I'd like to talk about epithelial cell-specific FRI1 knockout mice and adipocyte cell-specific FRI1 knockout mice. So we generated epithelial cell-specific FRI1 knockout mice by crossing FRI1 flox mice with keratin-14 Cre mice. And we confirmed the downregulation of FRI1 in the epithelial cell by immunohistochemistry and real-time PCR using epidermal seed. And interestingly, in this animal model, the altered epithelial phenotype of SSC patient was reproduced, such as the increased expression of keratin 6 and 16, and also the increased expression of IL-1 alpha and CTGF. So then we focused on the organ involvement of this animal model. So as you can see, this animal model spontaneously developed dermal fibrosis at the age of 3 months, as well as fibrosis and muscle atrophy in esophagus. And also, this animal model spontaneously developed interstitial lung disease with lymphoid aggregate. And also, we could detect the production of antibody in this animal model together with splenomegaly and lymph node swelling. And this chart summarizes the internal organ involvement of this animal model. So tissue fibrosis was exclusively detected in the skin, esophagus, and lungs, and lymphoid aggregate was predominantly detected in the lung tissue. So we tried to investigate the molecular mechanism which can explain this selective organ fibrosis in this animal model. So keratin-14 is expressed by stratified epithelia, which is distributed in the skin and esophagus. So as shown here, we could detect the increased expression of various molecules in the stratified epithelia of the skin and esophagus. So probably the expression of profibrotic molecules in the stratified epithelia can explain the selective organ fibrosis in the skin and esophagus in this animal model. And also, keratin-14 is expressed by medullary thymic epithelial cells. And Fly1 deficiency in MTEC result in the downregulation of autoimmune regulator AIR, and this abnormality leads to the atrophy of thymus and also impaired central tolerance. And we could confirm the presence of autoreactive T cells by adaptive transfer experiments in which we transferred T cells isolated from this animal model to RAG1 knockout mice. Under this situation, we could reproduce interstitial lung disease, but not dermal and esophageal fibrosis. On the other hand, when we crossed this animal model with RAG1 knockout mice, we could reproduce the fibrosis of the skin and esophagus without interstitial lung disease. So definitely the fibrosis of the skin and esophagus comes from the activation of stratified epithelia. So now, these results indicate that selective organ fibrosis in SSC, such as the skin, esophagus, and lung, can be explained by the activation of the epithelial cells, such as the activation of stratified epithelia and air downregulation in MTEC. So this is a new hypothesis. So we next focused on the adipocyte. So let's see the skin histology. So usually we can see a little bit smaller adipocyte in the lower dermis, which is surrounded with thickened collagen bundles. And usually in the early stage of SSC, skin fibrosis occurs in the lower dermis, which is adjacent to the adipocyte. So adipocyte seems to be involved in the development of dermal fibrosis in SSC. So again, we generated adipocyte cell-specific Fly1 knockout mice by crossing Fly1 floxed mice with atonectin Cre mice, and we confirmed the selective downregulation of Fly1 in adipocyte by immunohistochemistry and real-time PCR using isolated adipocyte. So here, we look at the skin histology. So this animal model spontaneously developed dermal fibrosis at the age of 3 months. And we also carried out double immunostaining for alpha SMA, a marker of myofibroblast, and perilipin, which is a marker of adipocyte. And we could see many double positive cells at the lower dermis. So these results suggest that the transdifferentiation of adipocyte into myofibroblast can explain the development of fibrosis in the lower dermis. So this chart summarizes the organ involvement of this animal model. So tissue fibrosis was selectively detected in the skin, but another important observation was the development of systemic vasculopathy in this animal model, such as in the skin, in the lung, and kidney. And the most important observation was that we could see the stenosis of pulmonary arterioles in this animal model, which get worse and worse along with aging. This observation is relevant to the pulmonary arterial hypertension-associated human SSC, the frequency of which is increased along with aging. So now we know that the development of dermal fibrosis in this animal model is attributable to the transdifferentiation of Fly1 deficient subcutaneous adipocyte to myofibroblast. But we cannot apply this theory to the development of systemic vasculopathy because, for example, lung tissue does not have adipocyte. And also, when we look at the expression profiles of adipokines in this animal model, we fail to detect any difference in the expression profile of adipokines. So we have to think another mechanism which can explain the development of systemic vasculopathy. Therefore, we next focus on the vascular remodeling system. So vascular remodeling consists of two distinct processes, such as angiogenesis and vasculogenesis. And angiogenesis is a vascular repair process which is mediated by the proliferation and migration of pre-existing endothelial cells. And this process can be assessed by retinal explant culture assay. And when we carried out this assay in this animal model, we could not see any difference between Fly1 Adipocyte mice and wild control mice, suggesting the no abnormality in angiogenesis in this animal model. On the other hand, vasculogenesis is a vascular repair process which is mediated by bone marrow-derived endothelial progenitor cells. And this process can be assessed by in vivo Madrigal plug assay. So we carried out this assay for these mice. And as you can see on the right side of this slide, so neovascularization was not observed in this animal model, suggesting the impaired vasculogenesis in this animal model. So vasculogenesis is mediated by endothelial progenitor cells, which comes from bone marrow. Therefore, we next look at the histology of bone marrow. So as you can see, we could see the atrophic adipocyte in the bone marrow of this animal model, suggesting the altered phenotype of adipocyte may affect the phenotype of endothelial progenitor cells. To address this issue, we look at the phenotype of bone marrow-derived mesenchymal cells because a part of endothelial progenitor cells comes from BMMSC. So as shown in this slide, the expression of alpha SMA was decreased while the expression of RGS5 was increased in BMMSC isolated from this animal model. And this phenotype was reproduced by co-culture assay between wild-type BMMSC and adipocyte isolated from Fly1 Adipocyte mice, suggesting that the altered phenotype of BMMSC at least partially comes from the altered phenotype of adipocyte. And as shown in this slide, the decreased expression of alpha SMA and the increased expression of RGS5 represent the predominance of synthetic phenotype of endothelial progenitor cells, which is characterized by increased production of extracellular matrix and increased proliferation and migration. And this cartoon summarizes our hypothesis explaining the systemic development of vasculopathy. So when the endothelial cells are injured, endothelial progenitor cells come to the injured blood vessels from bone marrow, and then these cells preferentially differentiate into fibroblast-like cells and induce the fibrosis of arterioles. So Fly1 deficient bone marrow adipocyte induce impaired vasculogenesis, which leads to the development of systemic vasculopathy in this animal model. So I talked about two new hypotheses explaining the systemic involvement of SSC based on the altered phenotype of epithelial cells and adipocyte. And the important thing is that basically these two hypotheses comes from the histological observation of the involved skin of SSC patient. So I think the skin is a kind of mine which is filled with clues to investigate systemic disease pathology. But the problem is, usually we just focused on the limited information of the skin. For example, in case of SSC, we just focus on the role of dermal fibroblasts, vascular cells, and immune cells. However, to know much more about the nature of disease, it's quite important to fight our sense of choice. So as a strategy to do this, we decided to generate various kinds of cell-specific Fly1 knockout mice. And indeed, this system works nicely. And eventually, we could encounter the new disease hypothesis based on the altered phenotype of epithelial cells and adipocytes. So I'd like to conclude my presentation by saying that when coming to a dead end, force yourself to see what you are not interested in. You may bump into a breakthrough. So I'd like to thank the organizer of IID meeting again for giving me this great opportunity, and I'm profoundly grateful to everybody who supported this study. So thank you for your attention.